Oh, Megan came. Wow, Megan. Thank you for coming. We'll get you out in time for your dinner at 6. How's that? Okay. So it's already 5:20. So we'll go ahead in interest of time. So first of all, I'm Manmeet Alwali. I'm a medical oncologist. I serve as the chair of the CNS Working Group. So thank you all for coming. You can feel free to come up front if you feel like, but if you can hear us clearly, just relax. It's an informal session. First of all, it's my distinct honor to introduce Dr. Justin Lathia, who heads the brain tumor research program at Cleveland Clinic. I personally had a distinct pleasure of working with Dr. Lathia for almost 12 years when I was at Cleveland Clinic. He's one of the smartest basic science researcher we have in the field of brain tumors. He is the leader or a principal investigator of PO1, over $10 million PO1, looking at sex differences in glioblastoma. And amongst a lot of other things he has done, he's been trying to study why males and females have differential outcomes in glioblastoma. So today he's going to talk to us about use of gliptins for targeting myeloid cells in male glioblastoma. Justin.
Okay, wow. I'll stand back a little bit. Manmeet, thank you so much for the opportunity to present. I'm going to keep this very informal, and I'm going to try to sort of keep this a little bit lighter. I know we're at the end of the day. So I want to briefly talk about sex differences in glioblastoma, because I think they not only do they have profound impact on the biology, but I think they also present a clinical opportunity. And I want to sort of, at a very high level, go through the things we've been working on in the laboratory. And a lot of what we've been focusing on is the myeloid lineage, which, as many of you can appreciate, is somewhat unique to the brain in that there's a huge myeloid influx as well as resident myeloid cells. And we've actually seen sex differences in microglia, and there's clinical implications in that, sex differences in myeloid-derived suppressor cells, which I'll talk about a little bit today. And then really, there's also sex differences in T cells. We just published that several weeks ago. And again, I want to end with this concept of, I think there's an opportunity to actually target male glioblastoma with gliptins. And I'll tell you a little bit about gliptins in a second. And then there's the disclosures. So, the question the laboratory has been asking for almost a decade is glioblastoma actually impact males and females the same? And the short answer is no. So here are incidence curves, whether it's all gliomas to the left or glioblastoma to the right. And there's two groups. There's a red group and the blue group. Is this P53 mutation? It's actually not. Is it environmental factors? It's not. It's actually biological sex. So this is just a demonstration that males get gliomas and glioblastoma more frequently. And what's interesting is if you actually look at the human population, I'm showing you a diagram of individuals dressed in white and black. From the left, 5 feet 0 inches, and the right is 6 feet 5 inches. So even a pedestrian phenotype like human height actually shows us a sex difference because the individuals dressed in black are biological males and the ones dressed in white are biological females. So as we think about the complexity of human biology, I think that these sex differences are actually embedded within who we are, actually within our DNA. And a couple of years ago, we published this in collaboration with Jill Barnholt Sloan, where we looked at the Ohio brain tumor study. And these are patients that have undergone standard of care. Hopefully you can appreciate that males actually have a worse prognosis. So think about what I just said for a second. Males get these tumors more frequently, and they actually do worse. And what's interesting is there's a lot of work in terms of the genetics. So take a look at that paper that was led by Josh Rubin at WashU, where there's actually differences in gene programs between male and female glioblastoma. And what's very interesting is embedded in that paper, there's a large-scale drug screen from both mouse and human GBM cells where temozolomide is actually more effective in females than males. So standard of care from a chemotherapeutic perspective is actually more effective in females than males. And obviously that's a huge problem when you think about what I just showed you. So if you think about sex differences in general, we've been very focused on this idea of sex differences in the immune response. So this is a very classic graph that a lot of people show. So on the left are autoimmune diseases. So if you think back to medical school, you probably learned about this, right? There's differences, for example, in lupus or multiple sclerosis. These are heavily female biased. And what's on the right is a male bias towards non-reproductive cancers. Now, the initial explanation for this was this is a direct result of poor lifestyle choices by males. But when you take that into account, epidemiologically, it still shakes out. And again, we're in the era of just coming out of a pandemic, and COVID-19 itself has shown a sex difference. So we've been interested in the myeloid-derived suppressor cell population. And this is work that was done by Mike Vogelbaum years ago, where he looked at age-matched individuals, and he actually found that the granulocytic subset of myeloid-derived suppressor cells was higher in glioblastoma patients. Let me go back a second. And what's interesting is if you look at other tumors like renal cell carcinoma, bladder, and melanoma, glioblastoma patients had a higher amount of these myeloid-derived suppressor cells. We've actually followed this up, and here's a graphical abstract from a paper we published several years ago led by Tyler Albin, who was a PhD student in the group. And what we did in our brain tumor patients is we looked either in the tumor microenvironment or in the circulation. And patients that had a good prognosis actually had limited amount of myeloid-derived suppressor cells between their first and second surgery in the tumor microenvironment. If we serially sampled their blood, there was actually either a stasis or a reduction in the myeloid-derived suppressor cells in their circulation. Now, if you look at patients that had a poor prognosis, we actually saw an enrichment in these cells in their brains, as well as in their blood as a function of time. And we used a technique called mass cytometry time of flight, or CyTOF, to reconstitute the immune system. And what we actually found was patients that had a good prognosis actually had a myeloid-derived suppressor cell phenotype that looked like the cells were differentiating. And again, this was work that was done by Alvaro Alvarado, who's now at UCLA, who was one of our PhD students as well. So we've got a situation where patients that actually do worse have more of these cells, not only in their circulation, but in the tumor microenvironment. So we started thinking about how do we target them. Now, I will tell you that this is not a precise way of doing it at all, but we actually found that low-dose chemotherapy, and as a paradigm, we used 5-FU, but for the clinical trial, we used capecitabine. In our mouse models, we could actually experience a dramatic increase in survival when we targeted the cells with capecitabine. And you can see that down below. So this is the human data down below. And all this is a demonstration of is the fact that we can give oral capecitabine, and as we serially sample the patients, the more dose we give of capecitabine, the less myeloid-derived suppressor cells there are in their circulation. And again, all of that work's been published, but you can imagine the challenges here clinically and translationally. This is a non-specific chemo. You know, we're in the era of precision medicine and targeted therapies, and this is all we've got. So obviously we've got to do better. So we've gone back to the basic science and we've asked a simple question. What are the differences in the myeloid-derived suppressor cell subsets? So up to this point, a lot of our work was classifying an entire population that's very heterogeneous and treating it all as the same, but we were interested in the subsets. And this is work that was published several years ago, led by Defna Bayek. And essentially what we found here in mice, as well as in humans, the myeloid-derived suppressor cells of the monocytic lineage are enriched in the male microenvironment. And in the females, there was an enrichment in the granulocytic lineage in the circulation. Take a second to think about that. What that actually suggests is that immune suppression is set up differently depending on biological sex. So as we think about targeted therapies and combination therapies, this has got to be taken into account. Now, we've taken this a step further, and I'm going to show you the data from this very in granular detail in a second, but we actually found that the monocytic lineage relied on adhesion programs to get into the brains of the mice. So there's an opportunity here to target the transit of these suppressor cells by altering adhesion programs. So the way we did this is we started in our mouse models and we transplanted tumors, and we started adoptively transferring different lineages. Again, this is all published, so I'll go relatively quickly, but it turns out that you can actually make tumors more aggressive in males when you actually give the monocytic lineage. So this was telling us that these cells were actually driving tumorigenesis. We used a technique called ATAC sequencing to look for regions of open chromatin. And what really struck us is a lot of the regions of open chromatin that were predicted for the monocytic lineage had everything to do with adhesion. And that makes biological sense because these cells have to get into the brain and stay in the brain. So we went after the integrin family of adhesion receptors. Hopefully you can appreciate that in our mouse models, You can see that the monocytic lineage used more of the integrin, such as beta-7 and beta-1. Integrin function can actually be read out by a conformational change. So we used a flow cytometry-based antibody to show that the activation state was much higher in monocytic versus granulocytic, according to beta-1. We've got a great neurosurgical team at the Cleveland Clinic. So with our colleagues there, we were actually able to get matched blood and a tumor from the same patient. And again, hopefully you can appreciate that the monocytic lineage here has just a lot more integrin beta-1. And then with single cell omics with Yifan at the University of Pennsylvania, we actually validated this all in publicly available human data sets. So this is really to just suggest that monocytic MDSCs, which again are higher in males, utilize adhesion programs like the beta-1 integrin program. We took it a step further and we did those adoptive transfer experiments, but this time we actually blocked integrin beta-1 that you can see on the top for 2 hours prior to adoptive transfer. And if you do that, actually the mice survive way longer. You do the same experiment with the granulocytic subset, that's the bottom left, you don't see a thing. And if you look to the bottom right, that's beta-7 and that doesn't give us a signal. So that's where we're at there. But I'll be the first to admit that targeting integrins is extremely complicated. There's a high degree of non-specificity. And as someone who 20 years ago did their PhD in the role of integrins in neural development, I can tell you that this is conceptually not a good idea. But can we learn anything from these data? So we went back to the ATAC sequencing data set, and our smoking gun was something called dipeptidase 4, DPP4. So DPP4, if those of you who have remember back to medical school, is very important in insulin processing and metabolism. So it's a huge target for diabetes. But it actually turns out it also interacts with integrin beta-1 on endothelial cells. So these are just the traces, and hopefully you can appreciate that there's a huge difference in the regions of open chromatin between the monocytic and granulocytic lineage. And if you look at the mRNA expression, DPP4 is quite a bit higher in the monocytic versus the granulocytic lineage. So we actually looked at patient MDSCs, and this is all bulk RNA sequencing. And one of our top hits was actually DPP4. And again, here's the single cell data to show it's relevant in humans. So DPP4, if you actually do simple bioinformatics, is higher in glioblastoma compared to lower grade tumors. And patients that have a higher amount of DPP4 in their tumor microenvironment actually have a poorer prognosis. So DPP4 obviously is a multifunctional protein. And what you're seeing on the left is all of the molecular mechanisms it's involved in, as well as the pathological states. But again, I just want to highlight that these are super well studied in humans in terms of the class of molecules called the gliptins. And you can see everything there to the right. So I think there's a real opportunity for translation here. And I want to sort of take you through what we're thinking. So it turns out that now we used the second gliptin there, the sitagliptin. So we use sitagliptin as a sort of paradigm. So we treated sitagliptin, and what we found was we could actually potently reduce migration of only the monocytic subset. One of the downstream signaling nodes to some of the gliptins are actually ERK. So you can see ERK phosphorylation is also reduced when we treat monocytic MDSCs with sitagliptin. If you actually give the mice sitagliptin, you can actually potently increase tumor latency by quite a bit. You can see that there. But what's interesting is if you do the same experiment in immune-compromised mice, the effect goes away. So we think that this is immune-driven. And you actually look at the percent macrophages. So the monocytic MDSCs can actually give rise to macrophages. And that process is severely blunted when we give sitagliptin. If you actually look at CD8 positive T cells, you don't see a major change. But what's interesting is that there's a weak activation signal, and that can be read out by, for example, CD69. We looked further at single cell RNA sequencing data sets, and you can see that there's a higher level of DPP4 on CD8 positive T cells. You can see that in the expression data on the left, as well as in our mouse models on the right. And what's interesting is if you look at other models of neuroinflammation, so here we used LCMV, it turns out that when T cells are So it turns out that if we inhibit DPP4 in vitro, we can actually activate CD8 positive T cells without altering PD-1 expression levels. So that to us is actually pretty exciting. So really, I'm going to kind of round this out and suggest that I think that we should be considering gliptins as an anti-GBM strategy against monocytic MDSCs. And again, these are the population that get into the male microenvironment. And as I told you, males get tumors 1.6 times more frequently than females. Right. So I would suggest that, and again, I've said part of this already, is that the tumor microenvironment is enriched in MDSCs. DPP4 is highly expressed by monocytic MDSCs. Gliptins can actually target DPP4, and in our mouse models, we get a decrease in survival. An increase in survival, a decrease in MDSCs. Also, an activation of CD8 positive T cells. Now, what's interesting is gliptins are very well studied in humans. I showed you that table. There's a variety of different flavors. But what's interesting is that very few have been done in the context of cancer, and none have been done with brain tumors. Now, there are some examples. So these are two papers that appeared recently, and they mainly use hepatocellular carcinoma and breast cancer as models. And I think at least as of this morning, there is a clinical trial using a DPP4 inhibitor for hepatocellular carcinoma. So what do we think this looks like? So obviously, we've been thinking about maybe starting with recurrent GBM and starting with males only. We've been a big fan, actually, ever since ManMeet was at our institution, for these window of opportunity trials, because as basic scientists, we want to learn as much as we can from the patients. So we have scenarios where we're suggesting we could give any gliptin for a couple of days prior to resection, and then interrogate the tumor microenvironment for immune cell changes, signaling alterations, and again, we've got candidates there, and then follow this with serial immune monitoring over time. And this is sort of the blueprint we use for the capecitabine clinical trial. So we're thinking the same thing. And obviously, primary endpoint would be safety efficacy, because this has not been assessed in this manner in recurrent GBM. And then obviously the secondary endpoint would be a reduction in the monocytic lineage and the increase in CD8 positive T cells. So again, it's still early days for us, and it'd be great to have engagement and feedback here. So I want to thank ManMeet for the opportunity to participate. Here are the folks in the lab. We've grown considerably over the last 11 years. And we've got great collaborators throughout the Institute, the US and the world. I want to thank the funding agencies. But again, I will say that, a lot of this has been a collaboration and mentorship from ManMeet, but also this has been a joint project with Defna Bayek, who has her own lab at the University of Miami. So I'm happy to stop there and take any questions.
This is really fascinating work. Question about gliptins, because I'm not as familiar with them. Like, I understand for anti-tumor treatments, there's only one trial so far for HCCs. What is the data for it in terms of, I guess, one, blood-brain barrier penetration, which I'm sure you'll talk about. And two, like, is toxicity or its safety profile, does it seem to make any difference by sex, genders, or not that you know of? Really curious about that information.
So let me answer your second question first. So in our preclinical models, because it's targeting the monocytic lineage, we have only used male mice. The signal is not as strong in female mice as it is in male mice, and that makes sense given what we've published. In terms of the blood-brain barrier penetration, this is giving me a chance to clarify. We actually think this doesn't need to get into the brain to work. Because remember, these lineage of cells is coming from the marrow. So it's marrow to circulation into the brain. Now, if you're inhibiting that process, there's a good chance that you'll be able to sort of get an effect. We've seen that in mice, and even in our capecitabine clinical trial, because the principle was the same. We know 5-FU doesn't get into the brain. We've known that for, 30 or 40 years, right? It's been a while. So I think that the mechanism of action here would be a peripheral targeting. So we're not as concerned about PK/PD in the brain.
Okay, thank you. I appreciate that.
So Justin, if you were to do a clinical trial with a drug surgery drug and build some of the correlatives, would you just do it in males or would you do it in females to see, if there is a greater, benefit in the males? Because otherwise the control part becomes an issue. Obviously, you could always do it without giving a drug where there'll be a differential effect. But I'm saying How do we measure the sex-based differential effect?
I know I had this crazy idea to only do a clinical trial in one sex in GBM.
I know.
But you know, I'm sort of open to any suggestions. I guess we could, you know, a trial could be written with the hypothesis that the magnitude of effect will be greater in males than females. I mean, that's something that we could, you know, it could certainly be written.
So for people like us, right, and there are limited people in the room, but people who are not in lab on daily basis, the differential effect that do you have a sense of prediction based on mice experiments? Is difficult to do because like canakinumab, we had a beautiful story.
Yeah.
Unfortunately, the drug went under. I went up to the CEO, Vas Narasimhan, and he couldn't give it to me because Novartis just disbanded the drug. But if we were to do a trial with gliptins, the reason why I'm asking is because, you know, there was a lot of interest in, for example, metformin, which is another anti-diabetic medication as a drug for cancer, right? Large trials, that some of the initial trials had shown a benefit. But when we did trials in GBM with our limited numbers, it didn't translate to any clinical benefit. So that becomes a challenge. So any thoughts on that?
I mean, if you're looking to induce wild speculation, I'm happy to give it to you. I think the short answer is I don't know. From a translational perspective, I don't feel comfortable coming to someone like you with a concept unless we see a signal in mice. I think we have to start somewhere. I think we can all accept that, but we know that there's a poor conversion rate from a signal in a mouse to a signal in a human. So we're very aware of that, but I think that's why if we can design studies with this window of opportunity in mind, we're learning every step of the way. And that may potentially allow us to stratify patients. You know, we have another trial that we're working on with another anti-MDSC inhibitor that, you know, we are now getting a sense that the amount of T cells in the tumor microenvironment at initial resection may serve as a biomarker for stratification. So I think we've got to do the first trial to figure out what's going on. And then I think we can get a little bit smarter. But I think the more data we can generate, the better from these clinical trials.
Okay. No, great. Thank you so much. Any other questions for Dr. Lathia? If not, thanks, Justin, for coming. Okay. I know you normally don't come to SWOG meetings, so we are very privileged to have you here, and we'll definitely discuss next steps, whether we do it as a small IST before we bring it to SWOG as a proof of concept, because I think that's what frankly will be needed. Yeah. But we can certainly have that. And it looks like a lot of your collaborators are now in Miami. So people are filing sunnier or greener pastures.
I was just waiting. I was waiting for that.
We even have Nima Sharifi, actually, who was on your slide. Okay, thanks for that exceptional talk. Next, we're going to move on. It's my, again, privilege to invite Dr. Tony Wang, who's a professor and a leading radiation oncologist in the field of brain tumors. And we are very privileged to have him here. He has got a very interesting dual immunotherapy concept that he would like to discuss with us. I did discuss this actually at the early rare tumor and therapeutics group, and I think there was some support, more than some support for those concepts. And we have Megan here who's going to help us drive this forward.
Perfect, thank you. And I also just want to add, there's actually, with respect to GBM concepts, I know there's one androgen deprivation concept that's kind of being floated around at NRG. It's by Chi Zhang. He's a radiation oncologist in Nebraska who presented the concept on that for newly diagnosed GBM. So thank you, Dr. Ahluwalia, for the opportunity to present this concept. This is a phase 2 study of atezolizumab and tocilizumab with RT in patients with newly diagnosed MGMT unmethylated glioblastoma. These are my disclosures, and they're electronically available. So most of us in this room probably familiar that glioblastoma is the most common primary brain tumor. It's very aggressive. And the median overall survival, even with radiation temozolomide, is approximately 14 months for all comers. But the benefit primarily with temozolomide has been shown to be within the subgroup of methylated MGMT promoters or methylated MGMT. So MGMT methylation, as we know, is prognostic for, with methylated MGMT, which is about a third of our patients with newly diagnosed GBM, the median overall survival is about 18 months. And with unmethylated MGMT, however, which is, over 2/3 of GBM patients, the median overall survival is approximately 12 months. And also MGMT methylation is predictive for the benefit of temozolomide. If you have methylated MGMT, temozolomide helps increase your median overall survival by approximately 8 months. But if you have unmethylated MGMT, your median overall survival is increased by less than a month, approximately 3 weeks, actually. So, this is a study of colleagues at Northwestern looking at the TCGA cohort using RNA sequencing and microarray data that demonstrated that interleukin-6 expression is associated with worse overall survival in glioblasts and gliomas in particular, both in RNA sequencing TCGA data, as well as in the microarray data as shown from these already well-published studies. Whereas low interleukin-6 expression levels are associated with better median overall survivals. And this is a study looking at anti-IL-6 therapy that helps show the reduced burden of M2 CD206 macrophages. When you give anti-IL-6 CSF treatments, it does help reduce the M2 CD206 macrophage expression levels. And this is a study from our colleagues at Penn using anti-IL-6 antibodies on genetically engineered orthotopic GBM murine models, where anti-IL-6 therapy shown prolonged survival in these GBM mouse model studies versus your standard control arms. And we also know that anti-IL-6, in the realm of immunotherapies, helps reduce CD68 myeloid cell infiltration. More importantly, also increases the tumor immune microenvironment by increasing CD8 positive T cell infiltrations with the help of anti-IL-6 therapies. So this is one study from our colleagues in Northwestern by Joe Lamano looking at anti-IL-6 as well as anti-PD-1 therapy in C57 BL6 mice, where they injected either anti-IL-6, anti-PD-1, as well as combination of anti-IL-6 and PD-1 therapy to find that there was reduction in tumor volume as well as improved survival in these animals who got the combination of anti-IL-6 and anti-PD-1 drug therapy. And this is one actually study that's older from China using U251 GBM xenograft models using tocilizumab, which is an IL-6 inhibitor, which helps promote apoptosis. And in their control group versus tocilizumab, the injection of the anti-IL-6 therapies help reduce tumor volume. And as shown on the bottom right, the IL-6 in the serum of these mice also dropped significantly with tocilizumab treatments. And again, showing from our colleagues in Northwestern, the combination of anti-PD-L1, IL-6 inhibition has been shown to exert additive anti-tumor effects in intracranial GL261 tumor-bearing mice. These are the combination of IL-6 and PD-1 to improve treatment survival in these animals versus just monotherapy or frankly just the control group. And I also want to point out that there's currently an ongoing NRG cooperative group trial. It's a phase 2 trial using atezolizumab, tocilizumab with reirradiation, in particular fractionated SRT, 8 grade times 3, in patients with recurrent glioblastoma. There's patients who are either surgically resectable or unresectable who will get a challenge of atezolizumab followed by FSRT of 8 grade times 3, and then continue maintenance atezolizumab until disease progression or death. So the concept here is that we propose is a phase 2 newly diagnosed GBM for patients with unmethylated MGMT to continue with radiation atezolizumab followed by atezotoci using the same doses as used in the NRG study. Primary endpoint is 1 year overall survival. Secondary endpoints include progression-free survival, as well as evaluate safety of adding atezolizumab and tocilizumab in combination. Other exploratory analysis include quality of life and tracking steroid uses, in particular with the dual immunotherapy study. This trial design mirrors design of multiple ongoing and completed studies, including Checkmate 498 and RGBN007 for newly diagnosed glioblastomas that omit temozolomide for glioblastoma patients without MGMT promoter methylation. As shown in both Checkmate 498 and NRGBN007, omitting temozolomide was not a barrier. Both trials accrued very well, and steroids are only given if clinically indicated, not empirically. It follows the precedent of certain other dual checkpoint inhibition studies. So following precedent of unmethylated MGMT, no temozolomide, no placebo, helps avoid T cell depleting effects of temozolomide in the experimental arm. And because these are unmethylated MGMT patients, this certainly would shorten the follow-up patients for these patients with
poor prognosis. So with that, I want to thank you for your attention and open any questions.
Any questions for Dr. Wang? If not, Megan, since you are here, I'm going to ask you the challenging question. I know SWOG, just like any other cooperative group, does not like smaller trials, right? But we are a rare tumor.
I have a portfolio.
Okay, good.
Okay.
That's the first hurdle. That's Megan. And the most important one. I know.
Yeah.
No, yeah, but what I'll present will be a sister concept, because the challenge is, very honestly, it's a dual immunotherapy concept. And we just had a trial that,
BN07, yeah.
BN007, which I spoke about in the morning session, that actually showed any lack of benefit, both in progression-free survival and overall survival. So if we were to go in with a concept for a randomized phase 2 trial, they'll just kill it. No matter how much, it will have a very difficult time getting a dual immunotherapy through right now.
Although, as you know, I'm on that study too. It was Ipi-Nivo, which is, the MOA is a little more, outdated versus like IL-6. So I think it's a little different, but yes, I agree. Both BN07 checkmate sort of has dampened our interest or excitement of IO for newly diagnosed GBM. But, yeah.
Okay, great. So Megan's given us a green light. So next thing we'll be working on a capsule and, okay. We have a relatively small program today, and I would like everyone to enjoy the evening or whatever we have. So I'll try to get people out of here. I did present this concept in the morning, and this is a sister concept to basically what Dr. Wang presented, because that was for unmethylated patients. This is for methylated patients, and it's a TRC102 combined with temozolomide now, because we know this is a patient population where we do get benefit. So, for people who don't treat brain tumors on a daily basis, temozolomide forms O6-methylguanine, 7-methylguanine, and 3-methyladenine DNA adducts when we treat patients with GBM with temozolomide. The 6-guanine methyl adduct is repaired by MGMT. And this is really the differential which Dr. Wang spoke about. So patients who have MGMT methylated tumors do not have this repair enzyme. So as a result, they get much more durable benefit with temozolomide compared to unmethylated patients, where the benefit is really 21 days. In methylated patients now, the benefit's around 6 months to 7 months. Now, but at the same time point, there are two other adducts that are formed, that is 7-methylguanine and 3-methyladenine, that is actually removed by the base excision repair pathway. And TRC102 is a base excision repair inhibitor that was founded in the lab of Dr. Stan Gerson, who was the Cancer Center director for Case Comprehensive Cancer Center, and now serves as the dean for the Case Medical School. And essentially, this is a base excision repair inhibitor that binds to the apurinic site created through the addition of MPG, which stands for N-methylpurine DNA glycosylase. And it's the first step in the base excision repair pathway. And work from Andy Sloan's lab showed that in orthotopic xenografts, if you combine TRC102 along with temozolomide, you actually got most durable benefit, and you can see a nice flattening of the curve there. So we tested this in a clinical trial that I actually got a career development award for. So I was a little bit younger then. I was in 2014. It tells you how much time it takes to do a trial through a cooperative group. You actually get a lot more gray hairs. So this is a phase 2 study of patients in first recurrence of glioblastoma. Primary endpoint was response rate, and CTEP had really put a Herculean task of getting a response rate of 30% in recurrent glioblastoma, which I think is almost unachievable, sadly. So anyways, we treated 19 patients. We did not meet the primary endpoint. However, our progression-free survival was 2 months, which is time to the next MRI. Our median overall survival was 11 months, which is the story of our lives in recurrent glioblastoma. Just to put this in perspective, there's not even a single therapy that has shown a single day benefit in recurrent glioblastoma. However, on the trial, I actually had probably 10 out of the 19 patients I personally treated. Out of them, 2 were exceptional responders. One had a progression-free survival of roughly 18 months. Another one had a 30-month progression-free survival benefit. And so we did a bunch of correlative studies that the combination was actually well tolerated. What was interesting for us was that in MGMT methylated patients, although there were only 5, and hence the confidence intervals are pretty large, median overall survival actually even in first recurrence was 30 months. That's pretty dramatic. So that's the first hypothesis, that now if you're getting a double hit strategy, you actually are getting benefit, which, from a disease biology makes sense, and from the data that I showed you. What we also found out was that we actually had an IRB. So we sent tissue over to Mike Beren's lab in TGen, who did whole exome sequencing and transcriptome. And what he showcased was that there was actually a DNA damage response pathway signature that we picked up. So 2 patients that I treated, actually I treated the third guy also, who almost got the best borderline benefit. So 2 exceptional responders, they have this gene signature that you're seeing on the right side. And then there was a patient of mine who actually got 6 cycles, but because our cycles are given every 28 days, he fell short of 180 days. So he didn't meet PFS 6, but he had a borderline signature. And we only had 7 patients on which we had tissue. So out of them, 4 were those who didn't do well, 1 had done borderline well, and 2 did really well. And the signature actually matched. So on the basis of this, we are proposing a phase 1/2 design of TRC-102 combined with temozolomide with newly diagnosed glioblastoma along with radiation. And we'll have 2 dose levels. One will be dose level 1, 75 milligrams per day, and then minus dose level of 50. And we will validate the DNA damage response pathway in a prospective manner. Now, when we have looked at the TCGA, this signature actually exists in around 10.5% of the patients. So very clearly, we had it in 2 patients, which was out of 20. So that's 10%. So it actually holds true. But in an MGMT population, that will roughly be 1/3, because only around 1/3 of all GBMs are methylated. So we are hoping that if we run this trial of around 40 patients or 50 patients, that we'll be able to get at least between 10 to 15 patients, so we can really truly validate this pathway. So this is what we are hoping to do. So the next steps is Dr. Wang and I and Dr. Venure has offered to help me. We'll work with Megan and to put together a capsule for the next steps for the SWOG committee. There is definitely support from the top with Dr. Blanky trying to be helpful. And I don't know, there are a lot of competing protocols, frankly, through the early rare therapeutic committee. There are not too many, to the best of my knowledge. So, and then I think these are different concepts than what are being floated through some of the other cooperative groups. So I hope, one of our strategies, although these are small, we can probably get them done in SWOG. We might reach out to NRG for support, because I've done the same favor to them. Whenever they floated a concept, I've provided them support from SWOG. So hopefully they'll reciprocate. Any questions for me? If not, thank you everyone for coming, and enjoy the rest of the evening.